Speaker, I think everyone knows, since probably everybody's got an email from him, is Kelin, and he will speak on persistent function-based machine learning for drug design.
Yeah, thanks, Constantine, for the introduction. So today my talk will be a little bit about application side. So the importance of drug design, I think need no further emphasis. However, this traditional way of drug design is not only time consuming, but also highly expensive. So currently it takes about 10 years and $2.3 billion to just bring one drug into the market. So recently, due to the great development in the data science, so this AI-based drug design and discovery area has begun to pick up momentum. And the underlying reasons for this progress can be really understood from like three major aspects. The first one is really about biomolecular data. So we have a huge amount of data currently available. For example, in this protein data bank, so currently we have about 178,000 structures. So to give a perspective, for every human being, we only have about 20,000 types of proteins. So here we have large amount of data for the protein structures. So this is for the crystal structure, the 3D structure. And then another more, gigantic amount of data is, gene data and this sequence data. So currently we have more than 220 million these gene sequences deposited in gene bank. And getting the gene information is become extremely cheaper and cheaper. So you see, in 1990, when people started this human genome project, at that time, getting the human genome information, which is about 3 billion base pairs, cost about $100 million. And it takes about 10 years to have this project done. But nowadays, some company can just get this whole thing done by using maybe like a cost you like $300. So the costs are decreasing dramatically. So that's also why, in this COVID-19 pandemic, we got a lot of like PCR tests that is tracking the gene information about the virus, if you have, mutation, alpha, gamma, this kind of things. So getting the information become more and more easy and resulting in a huge amount of data. And another thing is the computational power. So one of the example I really enjoy is this one. So in 1972, the Apollo 11 helped the human being landed in the moon for the first time. But at that time, the central processing unit for this Apollo 11 has only a RAM about 4 KB and a ROM about 72 KB. Nowadays, for any smartphone we have in our hand, so you see, no matter it's like Apple, Samsung, whatever, it easily can go to 8G or sometimes 16G or 32G. So what that means? That means the smartphone and everybody's hand is now millions of times faster than the central, the CPU system for the Apollo 11 that help us land on the moon. So that's the huge increase for the computational power. And then the next thing is really about the various learning models. So I don't need to go to the detail. Various machine learning, deep learning model has been proposed and really help make a lot of this highlights and fuzzy across the world. So from the mathematical point of view, for all these data learning model and the machine learning model, deep learning model, boil down, it's really about two things. The first thing is about data representation, and the second thing is feature learning. So whenever you have a data, what you want is to represent in certain form, certain mathematical form, and then from the mathematical representations, you want to generate certain features, and more importantly, you want to establish certain feature function or feature property relationship. For example, I think most of the people can get the joke here, the Obama and don't share the same facial expression. However, if you say, okay, can we let the computer find out all these images, for animal and human share the same facial expression? In order to achieve that, usually people have to done, again, following these two steps. First, you have to represent all the images in certain representation. For example, if you have an imaging, you can pick up certain representation point. At this point, characteristic point representation, or you can end up in certain, graph representation, or, maybe end up in certain meshes, and then certain, heat functions and certain eigenfunctions. All these things are just a certain representation of imaging data. And then from these representations, you need to come up with features that are related to the expression. And then you begin to establish certain feature function or feature property relationship. The same logic applied to all these AI drug design problems. So essentially, you need to first have a suitable representation for all these molecular data. So usually they are like, most popular is definitely the graph representation. So, and we heard about Herbert talk about the molecular surface or volume representation, and then there are certain density function representation from the electron density information of the molecules. And from these representations, there are actually a gigantic amount of features, or they call it molecular descriptor or chemical descriptors that has been proposed. Currently, there are several books just designed for, contains all these kind of descriptors. Surely some of these descriptors are related to the, physical, chemical, and biological property, but the majority of them, like 80 or 90% of these descriptors are really taken from the structures. So they have something called a 0D, 1D, 2D, and 3D. So 0D, they just count the molecular weight, how many atoms, how many bonds, and then they can get up into certain, graph. And then usually here they count a certain function group. It's just like a subgroup or certain local regions, group count. And then they go to a little bit of topology. So here, majorly, they refer to graph. And based on the graph, they have all kinds of different indexes. So this index usually are averaging between the distance between, several atoms, things like that. And then there are structural geometry. So they have really studied the surface information. And then they go to 4D. 4D basically means the protein are now and a certain dynamic. So they have a trajectory of proteins and then to take out certain features from them. And then they have something called fingerprint. A lot of them has been proposed. If you dig that, there's a gigantic amount of literature and works along this line. And one of the underlying reasons for these things is actually they have something called a quantitative structure activity relationship. Exactly what they do is trying to establish certain relation or function relationships between these descriptors and certain biological functions. So as a mathematician, when we see, okay, there are so many descriptors, so many fingerprints, is it possible that some descriptors or some of these features, this fingerprint are more fundamental, more intrinsic, and then maybe capture more general global informations that can be transferred from one system to another system. So that immediately lend us into the topological data analysis, because topological intrinsic invariant can really characterize this, intrinsic informations. I don't need to say too much about TDA here. A lot of the founders and the inventors of these areas are here. So when we study these systems, we are actually approaching from a different perspective. We are treating this persistent barcode information or diagram information from the molecular data as a fingerprint. So one difference is that our data are actually come from experimental result. So particularly from X-ray and NMR. So in this case, there are really no, not too much noise information there. Each bars there have its exact physical meaning. So at the beginning, we're trying to understand what are these bars represent. So we take this carbon-16 as an example. So it's made of 16 carbon atoms, and then they form this cage structure. So what we can see is that for battery 0, they have 60 bars, and 30 of them are shorter, around 1.38 astron. 30 of them are longer, about 1.42 astron. So astron is about 0.1 nanometer, which is 10 to the negative 10 meters. So here, these short bars actually represent exactly the carbon-carbon double bond, because double bond, they have like 2 pairs of electrons, so they're stronger. In that way, the carbon-carbon double bond are shorter, and these longer ones represent carbon-carbon single bond. So because they are comparatively weaker, so the distance between them are longer. And then we can check about the Betty zero bars. There are actually, 12 shorter bars and 20. Actually, here should be 19 bars because one is due to the combinations of the others. And the interesting thing is that if you check about all the molecular structures made from the covalent bond, you will only see pentagon and hexagon structure. Okay, so in these moleculars, we have exactly 12 pentagon and 20 hexagons. They are well represented in these barcodes. And if you go to the moleculars like protein, DNAs, when you check about the covalent bond reactions, you can only see pentagon and hexagons. There are usually no rectangular or other polyhedral structures you can find that. Okay, and then another thing is the Betty 2 bars. So here we use a Torres-Rips complex. So we have these short bars come from the hexagon, and then we have these long persistent bars which represent this cage structure. They form a sphere-like cage structure. They will contribute these very long bars. And later, what we can see is that we can actually just use this longest bar here, this longest Betty 2 bars, which roughly represent the sphericity of the structure. And then we can try to build up a connection between the inverse of these Betty 2 bars and something called a curvature energy. So I will not go to detail, but essentially they have a strong correlation with each other. When the moleculars are more sphere-like, they will have a lower curvature energy, and they have the more anisotropic, then they will have a higher energy in their formations. So after this simple carbon-16 examples, we dig a little bit more into the biomolecular system. So we have analyzed that our helix beta sheet, which are the two most important secondary structures. So these ones are actually directly related to the gram-CDNA, the ion channel. So if you have this ion channel structure, you take only the C alpha atom from it, you will see these very clear patterns. All the Betty zero, the bars will have the length around 3.8 astrons, and then all the Betty 1 bars will located at about 5 astrons. So if you want to check why all these Betty 1s come from, we can slice them out. You will see actually adjacent 4 C alpha atom, they actually form a very small channel structures. So if you have 5 of them, you have 2, so on and so forth. Similarly, if you want to see this big channel in this beta barrel, so you can take out the structure. Also, the C alpha atom, you will see the very long persistent bars over there. And then the adjacent C alpha atom all form these cycle structures around 5 astron, very similar to this one. And more interestingly, what we found is that for DNA structures, if you use suitable representation, you will immediately see there's a decomposition of the structure or separation of the barcodes. Some bars will come at an earlier stage of the filtration, the relatively short or at a more local scale. Some bars will come at the later stage of filtrations. So if you check about what's going on here, you actually see that each of the bars are actually corresponding exactly to the pentagon and the hexagon and the sugar ring structures in the nucleic acid. So you have A, T, G, and C. Again, as we said, the cycle structures are only made of pentagon and hexagons, and they will be exactly reflected here. And the global region characterize more of this. If you have 2 base pairs, they will have this, the loop region, and then you have minor and major growth, they will have a little bit more global or larger scale topological information. So these are represented here. So each of these barcodes here can really work as a fingerprint to identify different structures. So if you cap these bars, you will be able to say a lot about the structures. So in the beginning, we explore several different properties of the proteins. We have tried some protein folding. Yesterday, Professor Wei have already given some introduction about that. And then also some multi-resolution persistent homology analysis. So here what's going on is that if you say want to handling the virus, like the coronavirus or HIV virus, they have millions of atoms. So we have come up with some way to make it, transfer into density representation and then calculate barcode. And also we have handled with some cryo-EM data. So basically, there's some certain density representation and how it connected to the atomic resolution. So I will not go to too much detail of these things. I just want to emphasize two examples we have done recently that may be, more related to this. So first one is about this hydrogen bonding network. Okay, so it's really about the waters. So for each cell, actually 16 to 19% of its mass are made of water. So this water are H2O atom, right? So all oxygen and hydrogen, they have partial charges, so they can form a very complicated hydrogen bonding network. So essentially what's going on is that one part of the hydrogen atom will connect with oxygen atom from the other water molecules, and then they have this very complicated hydrogen bonding structures. And to make things even more complicated is that every molecular interactions or biomolecular functions or cell functions has to happen in a solvation environment. So we have to add in the salt, right, NaCl, sodium chloride into these systems, and these atoms, these ions will also interact with this, very complicated hydrogen network to make it even more complicated. So to check about some patterns of these things, we actually try some molecular dynamic simulations. So we analyze two systems. One is called sodium chloride systems. So and another one is KSCN. So basically two types of ions. So what we do is that we add these ions into the systems made of the same amount of waters, about 1,000 water molecules, and then we consistently add in the sodium chloride until it saturates. You cannot add two more because the deposit. So NaCl reach saturation about 5 more, and KSCN reach about 10 more, and we check about the corresponding the persistent barcode information for it. So we can see that for NaCl, they actually show this local clustering information. So there's again the separation of these local bars and the global bars. And what we found is that these local bars are actually come from the certain local clusters of these ions. They aggregate into the clusters, and then this cluster will be a little bit distance from the majority of the others. So it's just like a certain groups, and within this group they are very close to each other, but this whole group are far away from the general ions. And the KSCN do not have that. KSCN have very consistent, connected the network we call it extended ion network. So we again, more importantly is we check about the corresponding hydrogen bonding network. So get rid of the ions, only check about auto molecules, and then find out the barcode. So for NaCl, this is at a different concentration. KSCN also at different concentration. So already we can see that for NaCl, the change is not that dramatic for the hydrogen bonding network, but for KSCN, there seems to be a lot of changes or variations and barcode. To see it more clearly, we just add all these bars together to add it into this Betty curve. So this is the NaCl and different concentration. So we can see that the corresponding Betty curve does not change that much. There's only slight decrease of the peaks for this Betty curve for the NaCl system. But for KSCN, the situation are dramatically different. So this is a corresponding Betty curve. You see there's a huge decrease of peak value, and then the great increase in this a little bit more global regions. So that means that for KSCN, they are really forming a lot of very large circle structures in the system, in the global scales. And we have find out, they have these two patterns. And we talked with our colleague, they said, okay, this NaCl and KSCN are actually biologically represent two types of system. One is called structural breaking. So NaCl is just like in our day, if you add a lot of salt into the eggs, and you will denature the eggs. So this is called structural breaking. They will break the protein structures. And the KSCN are structure making. They are trying to alleviate the effect of this NaCl. So they are really representing these two different properties. So we check out another two systems, this what they call osmolyte systems, urine and the TMAO. So these two systems has been found to have the similar structure breaking and structure making mechanisms, just like NaCl and the KSCN. So we did the same thing. We increase the concentration. We fix the automolecular auto ions, automolecular in the system, and then we add in the osmolar molecules to reach saturation. And then we see what are the corresponding hydrogen bonding network looks like. So you can see that this is a result again for the Betti curve. The urine and the NaCl actually have very similar patterns. With the increase of the concentration, there's only slight decrease of the peak value. But for TMAO, we have also very similar result with the KICN. There's a huge decrease in the peak value and then increase in these regions. So these topological ideas can really help us dig out certain intrinsic patterns within these systems that has not been reviewed by other existing models. And another thing is this weighted persistent homology models that developed with my colleague, collaborator, Jiu. So what we do is that we try to introduce certain weight into the boundary operators. So you can, by introducing this, you have some extra degree of freedom to play with the data, particularly taking into consideration certain local effect and a certain interaction effect. Particularly for this interaction effect, say there are very different topological informations if you consider the structure and the effect or the interactions. For example, if we have this DNA structure, we just want to know the interaction between these two strings, then we can end up in this kind of bipartite representation, one from one string, another from another string. So in this way, this one represents the interconnection or the inner interaction between the two strings of DNAs. So this will be dramatically different than you directly do filtration on these type of structures. So using this kind of ideas to dig out more inner interactions or inner physical chemical reactions within the systems, we can really see more information. So one thing what we do is that we check about 3 different of these DNA structures using the traditional, so you just take the data and do filtration, you will not see the difference. You will not classify these 3 types. But using these interaction ideas, you will be able to classify them. And more importantly is that we can use them to really analyze the DNA structures and different solution environment. So DNA is here is in the pure water, this is in the liquid ions. They actually demonstrate very different patterns. So using the interactive persistent homology ideas, you can see that in the ion liquid, they have shown actually 2 different states. Okay, so that means that they will trap at 1 and these 2 different states during this dynamic process. But for the ones in the auto environment, there are only 1 state. And this is highly consistent with the experimental observations. So what we see is that using this type of analysis, we can really find out certain important patterns of the system. This is shows that the persistent homology can find out some information that cannot be observed by previous models. So later, what we do is that we try to combine the topological features with machine learning. So here is a general picture. Actually, there are already many work in this area. Sorry that if I'm not listing your work there. There's some booming areas in this thing by using the features from TDA and combined with machine learning. So the general flow, the flow chart for these things is that we have the input data, and then you have a certain representation, right? So here usually we use a simplified complex representation, and from there you have the barcode representation or barcode information, and then you extract certain features. This feature will be further input into the machine learning, either unsupervised learning or supervised learning, and then you try to have a prediction. So the Goes group actually have done a lot of work in this area, so and make a huge progress for this TDA-based learning models in drug design. So actually in drug design, there are various different steps, such as like the solution, how the drug can be sorbed into the water, how does drug transport the ions, how the drug have different toxicity, how it interact with different proteins. So all these things that are now various different data bands for you to track or benchmark your model. And actually TDA or topology-based model have consistently shown the best result than all the existing models. Yeah, to see how good is this, I think yesterday Professor Wei also mentioned about this D3R test. So currently in structural biology, there are two very famous or worldwide competitions. One is called CASD, one is called D3R. So this is just like the winter and the summer Olympics. So if you can get one medal there, it's already a very, very interesting thing, very innovative things. But you see, topological model can consistently deliver very good result and then get a lot of this first bench result comparing with existing models. These are really demonstrate the great power of these TDA-based models. So from a mathematical point of view, why this, the topological or TDA-based model can or has the potential to be so good? I think we can understand from several different perspectives. The first one is that traditional models, when biophysics or the chemistry people or biochemistry people, when they study the molecules, they usually use the graph representations. And then the edges are usually represented by the covalent bond. But the simplicial complex representation really has its great advantages that it has this simplexes which represent the high level interactions or many body interactions in this way. So let me try to figure out this for you here. So in this way, they can have these many body interactions instead of only the pairwise interactions as in the graph models. And another thing is that on the simplicial complex, various this invariant has been proposed, like a homology, co-homology, some of the spectrum informations. So this invariant measurement has great advantages because if the features are more intrinsic or more fundamental, they will have better transferabilities in machine learning models. So if your models, your feature characterize this intrinsic and highly abstract properties, then you can start it on one set of data and then easily move it to another. But if your feature is not this kind of intrinsic representations, usually they will have certain limitations. They may be suitable only for one set of data and cannot move to the other. So that's why this intrinsic representation is of great importance. And then is another thing is this filtration process. So physically speaking, the filtration process is important because it introduces this multi-scaled representations. So in biomoleculars, they have this covalent bond usually at like 1 to 2 angstrom, and then you have hydrogen bond, which is roughly about 3 to 4 angstroms, and then go to van der Waals, a little bit larger. Electrostatic interaction can go into 12 angstroms. So it naturally have this multi-scaled information, and the filtration process can really incorporate all these interactions at different scales, so that naturally match with the molecular properties. So based on this understanding, so recently we have proposed some models we call precision spectrum. So it's really about adding the spectrum model with the filtration process. So I don't need to say too much about this spectrum graph and spectrum simplicial complex. We have a lot of experts here. And another thing is that actually recently there are also something called a spectrum hypergraph. There are many different ways to derive the spectrum information from hypergraph. One area I think is very interesting is that they have this tensor representation. And based on the tensor representation, they can have the tensor spectrum information, and then they use that to characterize the hypergraph properties. So what we do is that we combine this information with the filtration process. So basically we started the evolution of the spectrum information during the filtration process. So if you have the filtration process, we can take, for example, the number of zero eigenvalues, then we end up into the Betti number. So here the multiplicity of zero eigenvalues are exactly corresponding to the persistent barcodes. And another more important thing is that we can check about the non-zero eigenvalues. So we have to check about different properties, maximum, minimum, standard deviation, and then some other like momentum properties, certain Riemann risk functions, and any other different type of functions. So essentially, this non-zero geometric information can be very helpful in characterizing the systems. Actually, we can already see that they have much more disjunctive, so that means they are more sensitive to the geometric information. So we have combined these things into the machine learning model and try to do some prediction. So one thing we check about is this protein ligand binding affinity study. So essentially, the drug will bind to proteins, and if they can bind it very strong, then they usually tend to change the properties of these proteins. Usually, it can help you cure disease or solve the problems. So predicting these binding affinities of great importance. Currently, they have established this what they call a protein PDB bind data bank. So you can bench up your models with this. All the other models, actually, we have systematically compared with nearly all the existing models. So for example, you can see here in this 2016 model, we have compared about 40 models. Many of them are very popular, like Autodog, ChemScore, and GomScore, widely used in the drug design communities. So another thing we have been played, or we have checked out, is the curvature. So there's recently this kind of oleo-rich culture and the form-rich culture, which is a discrete form of the curvatures on graph and a simplicial complex. So essentially, they have different properties. I don't think I need to go to the details. Very roughly speaking, the oleo-rich culture is related to certain, vast extent distances defined on the edges, and the adjacent atom, they have certain probability distribution, and then you can calculate the vast extent distance and then get into the curvature. And then form-rich culture is a more combinatory type of form. So you have the upper degree, lower degree, and then the adjacent atoms, and then from these combinatory representations, you can get the curvature on the simplexes. Very generally speaking, these curvatures can, characterize, even though the oleo curvature and the form-rich curvature are different forms, they are actually characterized very similar properties. So the positive ones are usually found to be densely packed clusters. So within a certain cluster or within a certain community, and then these negative curvatures are usually found in these linker regions and the bridge regions between the clusters. So what we do now is that not study the curvature of one structure, but instead checking the variation of the curvature properties during the filtration process. So we can systematically, evaluate all the curvatures, and then we take out the properties of the curvature we call persistent attributes. It can be certain statistical property, combinatory properties, or some other properties. And then you are checking the variation of this curvature information during the filtration process and use it as an input for the problems. So this is, again, combined with machine learning models tested on the data set. Again, it works quite well. So actually, as later we have found that we are slowly learning these things. So what we found is that actually, the data actually tightly connect with each other. So as we just see that from the spectrum, you have a 0 eigenvalue that gives you the homology information. And actually, the spectral model or the hot Laplacian model can be connected to the form-rich culture through this Bochner-Westland decomposition. So essentially, you have this combinatory Bochner, Laplacian, and then this is hot Laplacian. The difference between them is just the form-rich cultures. So in this way, they have, they're also highly connected with each other. They're just giving you the information from the different perspective. So this is what we do, and we roughly call all these things persistent functions. So they characterize the variation of the certain geometric and topological properties during the variations and during the filtration process, and they can be used as features for the learning models. So other than this feature, different persistent features, this multi-scaled features from the filtration process, we are also checking about different representations. So as we said, representation is one of the most important things in machine learning. So we have, previously we are basically dealing with the Ricci complex and the alpha complex. So working together with my collaborator, Professor Jie Wu, we have also checked about the hypergraph. So I think yesterday we have already had some discussion for the hypergraph. Essentially is that any set of these vertices can form a hyperedge, and then this hyperedge put together form a hypergraph. The good thing is that hypergraph have much greater flexibilities, but however, the boundary operator is not really that straightforward, can be defined in these hyperedges, because when you take the boundary operator, then you may end up in some situations that some elements are not existing there. So Professor Wu has done a lot of work here, proposed different version of the homology models, and we try to build up the hypergraph model based on the interactions. So you have the interaction between the ligand and ions, and these special interactions can be represented by these hypergraph models. And we also introduce this filtration process for the protein ligand interactions. So I will not go to the detail, but just saying that with the hypergraph, you have a much more flexible way of representing the interactions within the systems. And another more recent work is to check about other different types of simplexes. So previously, basically, we just play with Ricci and Ricci complex and alpha complex. But now we found out this neighborhood complex and Docker complex can be very naturally used in the biomolecular interaction process. So essentially what's going on is that if we have a protein and then we have a ligand, we just care about how they interact with each other, then these things can be represented just like a bipartite graph. So I have this part, the items from protein, this part, the item from ligand, and then I want to know how they interact with each other, right? So this is end up with this bipartite representations. And what we found is that the neighborhood complex from this bipartite interaction are exactly the Docker complex for this bipartite graph. So they're highly correlated with each other. So you see this one, each atom, say from here, the labels are all from the ligand part. And then for the ligand atom, say from here, all the labels are from here. So they naturally introduce into this Docker complex. So Docker complex, neighborhood complex are exactly the same, and the barcode, they have a very similar barcode when they're fully connected. And using these informations, we can also throw it into the learning models and try to do the prediction. So again, it works quite well. And I think I'm quite running out of time, so I just want to give a summarization what we want to do in our group. Actually, we are having this big umbrella we call the mathematical AI for molecular science. Essentially, we are interested in the molecular structures because molecular structures really demonstrate very interesting geometric topological properties. And many of these molecular structure data can be found from materials. There's a lot of very interesting materials, and in material design and discovery, a lot of good data that has also been built up, and also chemistry and biology. So from there we get this molecular data. And then we try to build up different representations. So essentially, when you have data, you can check, represent as a graph, as a surface, as density, as a certain combinatory information, or even some algebra representations like group or pole set. And from there, we try to dig out the features. Here, we majorly focus on all kinds of different invariant, geometric invariant and topological combinatory invariant, because these invariant, they are more fundamental, they are more intrinsic. Therefore, they will have better learning models. So again, when you learn from one system, this invariant, it can be easily transformed into another system for this invariant. So that's why we are directly focused on these things, and then just throw into all kinds of machine learning models, deep learning, digital learning, whatever, you will find a lot of that. So this is the bigger picture what we try to do within my group. So this my group members, Liu Xiang did a lot of hypergraph, and Junjie did the Ricci culture and spectral models, and I had some founder support. So that's all for my talk. Thank you.
Kelin, thank you. Can we, so again, as before, if people have questions, I think the easiest thing to do is just to put it up in the chat. Jürgen. Jürgen, you might be muted.
I just unmuted myself, so can you hear me now?
Yes.
Yeah, so thank you very much for very interesting talks that touches about many issues, perhaps more than I could possibly ask here. So I would like to ask a biological question. You talked about the structure of DNA molecules, but if I understand correctly, you just looked at them in some solution or so. Now, if you look at the DNA inside the nucleus of a cell, it has a very complicated shape. It's twisted, folded, curled, whatever, knotted. Would the quality of the data that one has there suffice to apply your methods, this TDA, to get some insight about this complicated structure of the DNA inside the nucleus of a living cell?
Yes, thank you for the question. So yes, so exactly I think the very first novel model that applied the topology into the biology is starting from the DNA knot. So it's check the knot structures for the DNAs. And recently, as just you mentioned, there's a lot of studies for the chromosome structures. So originally, they think the gene information is enough. So we have all the gene information, then we should have all the functions. And now they found out the foldings, what they call like super knot, or they have like this topological associated way, genomic apartment, they have all this high C data, the chromosome configuration capture data. They have all these things and try to understand the very complicated structure there within the chromosome structure. And then they believe these structures can, somehow say more about their functions. I believe some of they are already work for TDA within the chromosome data analysis. They have dig out some interesting information. Yeah, I believe there should be a lot of opportunities to play some of these models there. Even some cultural models, I believe it should definitely work very well there too.
Any other questions? Kelly.
Hey, thanks for the talk. That was actually awesome. It's awesome to see like TDA stuff, like outperforming benchmarks and stuff like that. It's always great. I was just wondering, so what are the size of the data sets that you're looking at here normally? Because I find, at least with I've started working with single cell stuff in TDA, and I find one of the big bottlenecks is that the TDA stuff often takes like so much longer to compute as the data sets get bigger. And I was just wondering what the scale of the stuff is with this molecular level.
Yeah, that's a very good question. So usually when we are dealing with these structures, they are not that large, and the filtration size we do not go that much. So previously, I didn't mention product here. So previously we have met with this problem when we are having this extremely large amount of data. We have like millions of data for this virus capsid. Okay, they're made up of this pentagon and hexagon ring structure. So what we do at that time is that we transform this transform this into a density representation. And then you have a density representation. You can use the volumetric data, and then you can we play with the what is the pursuits. Yeah, we use a discrete mode theory to calculate that. So in this way, you can bypass some of the calculations. But again, this is a big issues, and I believe there are some existing softwares at approximation. You have a large data, right? They can somehow do some approximation and reduce the computational cost. Yeah.
So sorry, with the density model, does that mean you're doing like a subsampling thing, or you're filtering some sort of density function? That wasn't quite sure.
So yes, it's just like a filter. So if I have an atom, right? So I have like a Gaussian distribution of the electrons. So I can transfer this point cloud data, which have a lot of coordinate, right, into a density representation data. That's just like ISO values in the space. And then I can use a relative cost representation. So that way I can calculate the most complex.
So this like, you make like a coarse grid structure or something and then use the density filtration. Okay, and that seems to work well, or it's like good enough.
Yeah, that can help to solve, alleviate some of the troubles.
Okay, cool. Thanks for that. Also, thanks again for the talk. It was really cool.
Wojciech.
Yes, thank you, Kelin. So I have a question, because you've done all these, a lot of experiments with real data. Have you found in any of those examples, that H2, the second homology, played a significant role?
Exactly. Thank you for the question. Actually, I don't say too much here. So you see, here we are dealing with the Betty-2, right? So again, we are checking about this longest bar here. So this longest bar basically represents roughly the size of this, carbon-60. So the problem here is that we have this, what do we call isomers. So they have the exact amount of the atoms, and they all form this sphere-like structure, but then not exactly the same. For example, here, I think this is carbon-40. So they make out 40 carbon atoms, but the shape are slightly different. Some are more regular or sphere-like. Some are more isotropic. So in this way, if you say, for example, I have these more isotropic ones, I will have certain regions that have a little bit more large curvatures. And to support these large curvatures, usually you need a high energy. So what we do is that we use the Betty-2 as a way to measure in the sphere-like of the system. So if the Betty-2 is large, it's more sphere-like. And then it's small, then it's more isotropic, right? Then you have these large curvatures. So in this way, we found that the inverse of these Betty-2 are actually linearly correlation with the energy. So you can use this one, easily build up a linear relation model and use it to predict the total curvature energy. So this cold curvature is related to how much energy you need to generate this structure. Okay, so you can find the experiment.
Very good. It's really great, really great. And this is not it's not accessible from H1 and H0, right?
Yes. So in this case, you don't need to count too much about H1 and H0. Yeah.
Very good. Thank you.
All right, we probably have time for one very quick question if somebody else has a question. And if not, then let's thank Kelin again, both for the lecture and for organizing the workshop.